Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. If you're watching these videos, you're either enrolled in my class or you stumbled across them in YouTube land. If you're enrolled in my class, these videos are intended for you to learn the material the way that I want you to learn it for now. Especially early on in part one, I very much simplify some things. We'll add the layers of complexity later on. If you're not in my class, make sure that you learn this material the way that your instructors want you to learn it. There may be more detail, less detail, and other topics covered. But if you find these videos helpful, hit like and subscribe, and you will get notified when new videos are loaded up. You know, I'm an analog guy in a digital world, so these videos are all shot in one take. If I sneeze or cough or whatever, I drop something, I gotta pick it up. I'm not editing that out. So today's video is sort of a continuation of the one that you probably should have just watched. We just completed DNA replication. Now, the reason that DNA is important is that DNA is the instructions on how to assemble proteins. Excuse me. So, let's say all of these markers that are lying up here represent amino acids floating around inside the cell. When you eat chicken or plant material or whatever you eat, inside the cells of those living organisms are all the proteins. We digest the proteins into the individual amino acids and send them to our cells. And then our cells, when we need a protein, we have to assemble that protein by putting the amino acids in order. Now, let's say for all my purple markers, right now I only have 3, but let's say I have a bucket here that has 10 to 50,000 of these. I mean, hundreds of thousands of these purple markers, all in the purple bucket. Let's say I have a pink bucket, and in the pink bucket are hundreds of thousands of pink markers and brown markers. Let's say I have 20 different buckets, 20 different colors, 20 different types of amino acids. I can have thousands and thousands of one amino acid. I can have thousands of these purple markers, okay? All in a purple bucket. I have thousands of pink in a pink bucket, thousands of red in the red bucket, thousands of blue, and I have 20 different choices, 20 different colors. If I told you, I need you to take some markers and I need you to assemble them all by sticking them together like this into a certain order and a certain number. I have that in my head, how many long I want my chain of them to be, and a certain order of colors. If I asked you to go ahead and assemble that for me, take my markers and stick them together in long chains so that by the time we're done, we have a certain length and a certain number of amino acids in our protein. And I'm thinking of a number, somewhere between 40 and 500. And I asked you to assemble them. And I said, go. How long would it take for you if you were guessing of how many and exactly the order? How many combinations and permutations of this could we come up with? It would be millions, right? How long would it take for you to figure out what I'm thinking? It would take forever. Life doesn't have forever. Things have to happen randomly. Now, oops, I'm dropping my markers. Now, I'm going to grab something. Let me step off screen. Let's say I have 2 books. Each book is a chromosome, a volume of information. And on every different page is the instructions on how to assemble a different protein. So this book has 107 pages. So there's 107 different proteins or 107 genes on this chromosome. Does that make sense? I hope so. On this chromosome, in this book, I have, sorry, let me get to a page number. I have 172 pages. So I have 172 genes on this chromosome. And every gene tells me how many amino acids and the exact order that I want them in. Now, If I need to make a protein, I need the instructions on how to assemble that protein. So I would go to that gene. Let's say it's on chromosome 2. And let's say on chromosome 2, it's going to be gene number 105 that I need built. I need to build that or turn that DNA sequence into a protein. Then what I could do is I could take that page and I can make multiple copies of it. So these are blank sheets of paper, but let's say I have 40 copies here of gene number 105 on chromosome 2. I could send each copy to a different ribosome, and that ribosome could gather all the amino acids that it needs, and it will know exactly what order to assemble them in. And so what I've done is I've taken a specific gene. I don't need to copy all of the DNA and then tell you which particular gene I need you to produce. I can go to the gene and I can make a version of that gene, like a photocopy of a page, send it to my ribosomes, and my ribosomes, multiple ribosomes at the same time, can all be building the same protein. Because when a cell needs a protein to perform a function, it doesn't just make one protein, it makes hundreds and thousands of these proteins to perform all these functions at once, or to be secreted and sent to another cell. Now, all of the DNA is housed inside the nucleus. That's where all the chromosomes are. So let's say the owner of our factory, our protein factory, has all the chromosomes inside his office, the CEO's office. That would be the nucleus. And let's say the instructions are all written in a language that the ribosome doesn't understand. And the ribosome doesn't have access to the office, doesn't have access to the nucleus. So when I need my factory to produce a specific protein, the ribosomes don't go into the nucleus and read the DNA. Enzymes inside the nucleus will find the specific gene on a specific chromosome, uncoil just that section of the chromosome, and then rewrite the series of bases, all the A's, T's, G's, and C's, into a language that the ribosome can understand. Ribosomes don't understand the letter T, thymine. They do understand uracil. So as I rewrite the piece of DNA, instead of T's, I use U's for uracil, so that when the ribosome gets the instructions, it can understand them. I'm going to give you a setup for what I'm talking about. That's what part of this is. But look, I'm going to write a bunch of letters across the top of the board here. I don't need you to copy this. I need you to understand what I'm doing, so pay attention. Okay? Let's say I have some letters here. If I asked you to start reading this 3 letters at a time and try to find the abbreviations in Spanish for a color, a 3-letter abbreviation for colors in Spanish, that doesn't amount to anything. That amounts to nothing. So as we go down here, I don't see anything until I get to here. If I start reading here, what I get is rojo, naranjo, azul, azul, moreno, negro, azul, rosa, naranjo, azul, verde, rojo, rosa, moreno. That's my end. This is a gene. The whole thing would be one part of a chromosome that could be thousands more letters long. And if I started right here with, I don't know, Then I would have green, pink. That would be another gene, starting a second gene here. So there is some pieces of DNA that don't seem to make sense. And what they do say, sort of, is, hey, we're about to start, and this is the end of a gene. Now, if I asked you to take those instructions and put these markers in a specific order, but you don't understand Spanish, then what you need is someone to come in here and find the gene that you're interested in and rewrite the gene into a language that you can understand. And so what we would do is we would start to do this. Red, orange, blue, blue, brown, black, blue, pink, orange, make that a G, blue, green, red, pink, brown. So now I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 amino acids, and I know the exact order that those amino acids are. The molecule that is in purple is my DNA. And what I do is I transcribe DNA into what's called messenger RNA. That's what this molecule is. It's going to be called mRNA. The M stands for messenger. And it's called messenger RNA because this message is going to go to the ribosome, and the ribosome is going to read it and go, oh, okay, let's see. I need a red marker first. I need an orange marker next. I need a blue one next. I need a blue one next. And then I need a brown one. And then I need black. Then I need another blue one. I don't have another dark blue, so I'm going to use light blue. Then I need pink. Then I need another orange. Then I need another blue one. I'm running out of blues. But I'm going to stop there, but you guys get the picture. I could build a series of amino acids into a protein using the messenger RNA. I'm the ribosome. Each marker is an amino acid. The whole series of them is a protein. When I get to the end, I would have 14 amino acids in a very specific order. There's no mistaking. And then that protein folds up and does its job. So the process of rewriting DNA into messenger RNA is called transcription. And this is a definition you're going to need to know. Transcription. Let me use a little bit darker color, just in case that doesn't show up very well. This is the process of rewriting DNA into messenger RNA. Two other things you need to know about this. Number one, it occurs in the nucleus, because that's where the DNA is. It is performed by the enzyme called RNA polymerase. And DNA polymerase and RNA polymerase are actually complexes of several enzymes, but we're not going to get into that level of detail. So this is exactly what happens. During transcription, something tells the cell, we need this protein now. And there's actually another structure that says, hey, by the way, do this. There are other proteins that bind, and we're not going to get into operons and a lot of complex biology, but you may have done that if you've had some biology before. But something says, transcribe this gene now. So our translator, which is called RNA polymerase, or transcriber, is going to come in and start reading it and rewrite the DNA into messenger RNA. Now, we don't use these letters. We only use A's, T's, G's, and C's for DNA, and we use A, U, G, and C for RNA. But ultimately, once I get done, the RNA polymerase will peel this off and then release it. It will be sent out of a nuclear pore to the ribosome. The ribosome gets the message. It knows which end to start. There's some ways that that happens. And it starts reading. And it starts assembling the protein. So those are the steps of transcription. And I'm going to kind of draw them out for you. But that's exactly what's happening. Now, let me erase all this, and we'll see how it really happens with some DNA. Okay? And I'll give you some more definitions, and I'll tell you what I can and might ask you on a lecture test. Now, so, here's what I want to get across to you guys and gals. If I were going to take a cell, and inside the nucleus are all the pieces of DNA, I'm going to cut this cell right here. And I'm going to make this edge of the video screen inside the nucleus. I'm going to draw a nuclear envelope, and way out here I'm going to have all the cytoplasm so we can see what occurs where. Now, so I'm going to have a coiled up DNA molecule with all the bases in here. A's, T's, G's, and C's. You guys are familiar with that. Essentially what's going to happen is I'm going to uncoil and unzip at a gene. And RNA polymerase will do that. It uncoils and unzips a double-stranded DNA molecule at a specific gene. So as RNA polymerase binds, it's going to start reading one of the two strands, ATC. Let's do it this way. TAG, CCA, GTG, CTA, TTG, right? And then these get stuck together. On this strand, I'm going to have ATC. I want to make this a C. I'll make that a G for now. GG, T, C, A, C, G, A, T, A, C. So I've actually unzipped the strands and uncoiled them and unzipped them, exposing a series of bases. And further up here, everything's still stuck together. Now, what has done this is a molecule called RNA polymerase. RNA polymerase is going to bind to one of the two strands. I'm just going to put RNA pol. As it binds to this strand, it can bring in complementary bases. And it's literally going to do it almost exactly like DNA replication. The only difference is RNA polymerase will not bind a T here. Wherever there should be a T, we're going to put a U. So it will bring an A here, a U here, and a G there, and string them together and bind them. And then over time, I'm not going to do this in a series of steps across the board, but over time it will switch down to the next triplet, and it has strung these bases together. Let me write that a little neater. And then it will bring in the next three bases, G, G, U, because there should be a T there. And it will string that all together, and then it will shift down and read the next triplet, C, A, C, and stick them all together. So over time, it's doing the same thing that DNA polymerase does, but it's building a molecule of RNA, so it's called RNA polymerase. And it doesn't completely unzip them because we're not copying the strands. It only unzips the gene that we need. It starts reading the triplets and adding the three bases on the RNA molecule. By the time it gets to the very end and it reaches the stop codon, it will pop off, and the entire RNA strand will get peeled off, and will be sitting like this: AUG, GGU, CAC, GAU. And I'm actually going to, just for illustrative purposes, put this on there again, and then I'll just throw another random one on here. Okay? I'm not copying my strand exactly, but I want to demonstrate something. Now, all of this is occurring inside the nucleus. This is the nuclear envelope. And that opening would be a nuclear pore. And our messenger RNA is going to be sent out the nuclear pore into the cytoplasm. So there's a series of steps that happen here. RNA polymerase will bind to, uncoil, and unzip a gene. It will begin reading triplets and adding complementary bases. It will shift to the next triplet and add the next 3 complementary RNA bases. It shifts to the next triplet and it repeats the process till it gets to the end of the gene and stops. We peel off the messenger RNA, send it out the nuclear pore, and then it's going to meet with the ribosome. Now, once my RNA molecule is outside the nucleus, so I'm going to rewrite this, and I'm going to rewrite it right here. So we send it out to the ribosome, and our series of bases is going to be AUG, GGU, CAC. I have GAU, CAC, and CUC. Obviously, it would be several hundred bases long, but nonetheless. Now, here's what happens. Our ribosome has 2 subunits, and it can read up to 6 bases. But a series of 3 bases on mRNA that codes for 1 amino acid is called a codon. You need to know that definition. A series of 3 bases on a messenger RNA molecule that is the instructions for 1 amino acid is called a codon. So I have several codons here. I actually have 1, 2, 3, 4, 5, 6 codons here. Okay. The ribosome can read up to 2 codons at a time, and what it does is, as it reads the first codon, it needs to bring in a specific amino acid. Now, if I step back to the example I was giving you a little while ago, I got all these buckets of markers up here, and you're at your desk out there working as a ribosome. I can't have all of y'all running up here, bumping into each other, grabbing markers out of buckets and running back. What if we had people who were transporters? And these transporters would transfer or carry the marker to you. And here's the rule. If someone has a blue shirt, they can only carry the blue marker. If someone were to have a red shirt on, they can only carry a red marker. If you're wearing a purple shirt, then that transporter can only transfer the purple marker to the ribosome. Now, as a ribosome, you could be sitting at your desk, and if you get to the first one, and this means the red amino acid, they're not called red and blue and green. They have names like leucine, isoleucine, threonine, serine, tyrosine. While you're reading your instructions, you could go, okay, I need a red one first. Hey, you in the red, come here. Give me your amino acid. And the transporter, called transfer RNA, will bring you the red amino acid, give it to you, and then come back to the red bucket and pick up another one and just wait for someone to say, I need a red amino acid, and then they deliver it. They just run back and forth delivering red amino acids from buckets to ribosomes. So the ribosomes could sit still and sit here and build their amino acids into a protein. I hope that makes sense, because that's exactly what happens. Now, when we eat DNA and RNA, we digest it into the nucleotides, and it actually, the components of a nucleotide, our cells absorb them, assemble nucleotides, and I got hundreds of nucleotides floating around in here. And those nucleotides would include things like A's and U's and G's and C's, as well as T's, but we're doing RNA. Now, the transport molecule that's going to be carrying the A around is going to have a whole bunch of bases. It is a piece of RNA. It's got A's, U's, G's, and C's on it. And I have all these little letters here. But in one part of the molecule, 3 letters will stick out more than the others. And this area right here is called an anti-codon. It is a series of 3 bases on a molecule called tRNA. This molecule is called tRNA, and it's called tRNA because it's going to transfer this amino acid to the ribosome when the ribosome needs it. How the ribosome knows which amino acid to put there is because this anticodon is going to match exactly with this codon. So there's only one tRNA that's going to have these 3 bases here in this area sticking out. And if these are the 3 bases, if I had another one of these, if I have another tRNA floating around that has UAC as its anticodon, it's always going to be carrying that particular base or that particular amino acid. I'm not going to put an A there. Let me put AA for amino acid. Maybe that's the code for the blue amino acid. Okay. Now, I actually started confusing 2 things. So when I have a series of bases together that forms a transfer RNA, the T stands for transfer, it's going to transfer this amino acid to the ribosome for assembly of a protein. And the reason it has this particular amino acid is there's a chemical bond formed based on the structure of this amino acid and this series of bases. And at one end, the anticodon is always going to have that same series of bases. Now, if I have another tRNA molecule here, and the anticodon here is going to be GAG, then that might mean a specific color amino acid or specific amino acid. If I have another tRNA floating out here, and it's going to have GUG, then that might be a different amino acid out here. Like, I don't know, let's do the red one. And if I have, let's do one more. I have another tRNA here that has CUC here. It might carry the black amino acid. So I have all these tRNAs floating around inside the cell, and a specific anticodon on a specific region here means it carries a specific amino acid. Now, when the ribosome binds to the messenger RNA, it can actually start to pull in some of these anticodons and match them up. The only one that base pairs with this using the base pairing rule is going to be whatever has UAC here. If I stick that anticodon on that codon, then I can only get the blue amino acid next. And because it can bind 2 at a specific time, and all of this has been figured out through chemistry and biochemistry and physics, there's only one particular tRNA that can bind there. And I don't have that series here, but if I had a tRNA that has CCA on it, that might mean that it's going to carry the pink amino acid. So guess what happens? Only the pink amino acid can go there. When these amino acids are held close together like this, they will break this bond, and this blue amino acid will form a bond with the next amino acid. And what happens is the ribosome is going to shift down. And when this whole thing shifts down, it releases this tRNA. Now the ribosome is reading these 6 bases. It's still holding on to this tRNA, which is holding on to that amino acid, which is chemically bound to that one, and it brings in the next tRNA that has the matching anticodon, which is going to be GUG. And I don't think I did one here, but let's say, oh yeah, I did. So that means only the red amino acid could go next. There's no mistaking. And then I'll break this bond, link these together, shift down and release this, and then it reads the next 3. And there's only one that will sit here, CUA. And I don't have one that has CUA, but let's just pretend that that's going to be this amino acid. It will bind the 2 amino acids together, release this tRNA. The tRNAs will go pick up another amino acid and run around looking to deliver it. And as the ribosome shifts down, it will bring in GUG. Well, that tRNA means always the red amino acid goes here. It binds, shifts, and eventually it will read the last one, which is going to be GAG, and that's going to be this color amino acid. As it gets to the end and is done shifting down, it falls off, releases all the tRNAs, and now I have my amino acid sequence. I have my protein, blue, pink, red, green, red. So the series of bases, the codons on my messenger RNA, tell the ribosome which amino acids go next. Those codons are assembled by using the series of bases on our DNA strand. By the way, the strand that is being read is called the template strand. The other strand is called the coding strand. And the reason it's called the coding strand is because its bases are the code or match the amino acid, match the bases in the mRNA, except for the D gets turned into a U. There's a lot of really cool, interesting stuff I could go into. Here's what I need you to know. I need you to know that this process of rewriting DNA into messenger RNA is called transcription. It occurs inside the nucleus. RNA polymerase will bind to the gene, uncoil and unzip it, bring in the associated RNA nucleotides, and form a strand of RNA molecules called messenger RNA. When it gets to the end, it falls off. The RNA gets peeled off, so to speak, and sent out a nuclear pore to the ribosome. When the ribosome gets the messenger RNA, it starts to read codons, 3 bases at a time. And as it does that, it brings in the matching tRNAs with their matching anticodons, which transfer the amino acids. And we assemble a protein that way. And every time I transcribe this gene into this messenger RNA, I will always get that protein built. There's no mistake about it. Now, there's a couple of other definitions that I want you to know. I want you to know what a codon is, a series of 3 bases on a messenger RNA that codes for a specific amino acid. An anticodon is a series of 3 bases on a tRNA that tell the tRNA which amino acid to carry. It also matches with the base pairing rule to a specific codon. The process of turning the messenger RNA, physically building or assembling the protein using messenger RNA, is called translation. I'm not going to write out the definition, but I'm going to tell it to you the way that I want you to know it. Translation is the assembly of a protein, 1 amino acid at a time, that occurs at the ribosome as the ribosome reads the messenger RNA. So translation is the physical assembly of a protein, amino acid by amino acid, performed by the ribosome as it reads the messenger RNA. tRNAs are the molecules that transfer the amino acid to the ribosome during translation. So a tRNA is an RNA molecule that transfers amino acids to the ribosome during translation. So essentially, here's some things that you need to know. Protein synthesis is the process of building proteins using the instructions of a gene. Protein synthesis involves two steps: transcription, the rewriting of DNA into messenger RNA inside the nucleus, and translation, the physical assembly of amino acids into a protein that occurs at the ribosome. And translation occurs at the ribosome in the cytoplasm. The molecule that transfers amino acids to the ribosome are called transfer RNAs. The three bases on a messenger RNA that code for a specific amino acid are called a codon. The three bases on a tRNA that code for a specific amino acid is called an anticodon. The molecule that copies, or not copies, that rewrites DNA or transcribes DNA into a messenger RNA is called RNA polymerase. And when we separate the strands, whatever strand is being read by the messenger RNA is called the template strand. The other strand is simply referred to as the coding strand. I can ask you any and all of those definitions. So make sure that you start understanding a lot of these definitions. I'll have a worksheet on this, and you guys can fill in the answers for it and practice that stuff. One last thing I want to show you, and then we'll wrap this video up. Okay, let me erase this. These are sort of, very popular or famous biology questions when it comes to this stuff. Which of the following molecules transfers or carries amino acids to the ribosome during translation? That would be tRNA. A series of three bases on a messenger RNA that codes for an amino acid is known as a blank, and you would say codon. Okay, now, let's say I give you two DNA strands. Here's a possible question. For DNA replication, what if I told you, here's a single-stranded DNA molecule. Please give the order of bases that would be found in the complementary DNA strand. Then all you would have to do is the base pairing rule. And it would be TTA, GTA, TGC. And you start checking answers. Answer A, answer B, answer C, answer D, answer E on a lecture test. And let's say A starts with UUA. Well, you know immediately that's wrong because we're going to be copying or replicating this DNA molecule. We want to know what the complementary strand is. If answer choice B starts with TTA, and then it has GTA, and then it has TCG, that's not correct. So this one would not be correct. If answer choice C has TTA, GTA, TGC, and this one has AAU, and this one has AAT, you could look here and go, this is my answer. That's the only one that matches. So on lecture tests for DNA replication, we can ask you the definition, where does it occur, what is the enzyme, or I could say, here's a single-stranded DNA molecule. What would be the correct order of bases in the complementary strand if we replicated this DNA molecule? And you could do that. Now for protein synthesis, it's different. For protein synthesis, I would have to give you two strands. And I would have to say, for example, coding and template. I have a coding strand and a template strand. And my coding strand is going to be AAT, GCA, CTC, GAG. Let's just say that's all I give you. The template strand is going to have the exact opposite. Now, I said, what if the question reads, Below is a double-stranded DNA molecule. With the information provided, please give the correct order of bases on an mRNA molecule if we transcribed this piece of DNA. You have to know this. I need to use my template strand. And I can go AAU, GCA, CUC, GAG. You could go to the coding strand and just replace the T's with U's and it would give you the same answer. And then you would have answer choices A, B, C, D, E down here. And you have to find the one that reads this. So you would actually have to know, I used the template strand, that would be the series of bases. If you use the coding strand and you did this, you would get the wrong answer. And you would find the correct answer. Let's say it's answer choice E and bubble that in. So you need to know the definition of DNA replication and all the stuff that we did with that. You need to know the definition of transcription and translation. You need to know which enzyme performs transcription. You need to know that we rewrite the DNA into messenger RNA. mRNA leaves the nucleus through a nuclear pore, binds to a ribosome. The ribosome will translate the messenger RNA into a protein. The way that it knows to add the bases, which base goes where, is that the ribosome will match the anticodon on tRNA to the codons on the mRNA and assemble the protein. That's all translation. Somehow, some way, I know this is a very difficult and confusing concept. It's far more complex than this, but I'm trying to simplify it as best I know how. So I hope you learned something from it. I hope you had as much fun as I did. Watch the video over and over and over again. And if you don't like my video, look up other DNA replication and protein synthesis videos on YouTube and see if you can learn something from it. Read the textbook and then practice doing it, writing out the definitions, doing the process. Do it till you can't stand it. Do it till you understand it. Then do it 5 more times and then try to teach it to a 7th grader. If you can do all that, you'll make an A on the test. Hope you had as much fun as I did. See you in the next video.